So today the story is mainly on the gene called DEMETER, which we named after the Greek goddess of agriculture. So mainly this gene regulates gene imprinting and is a master control of epigenetic, and it's an essential gene that is required for seed development. However, this gene only expressed in the central cell, so it's one cell. So that's why a lot of research was done by using other creative work, because we cannot get to that particular cell type easily. And that's also why the research progress has been quite slow. However, the discovery that has been revealed by studying this gene has been very important in our understanding of epigenetic process. So what is epigenetics? So to me, this is the simplest way of looking at epigenetics. So you think of it as a switch that you can flip on and off. So the DNA is going from the more open euchromatic state by a few simple modifications and turn into a more compacted heterochromatin state so that the gene is not active. And the reason it's not active is because its environment is compacted, so all the other DNA modifying enzymes like transcription factor or other things cannot easily get to. So switching back and forth between these two states is regulated by developmental cue, environmental stress, or maybe sometimes just spontaneously occur. Some of the modification like DNA methylation, histone methylation, and acetylation, etc., regulate this transition between on and off stage of the epigenetics. So we see the epigenetic all the time in our daily life. So these are just a few examples that I put together. So this is a maize kernel pigmentation. This is mainly due to a transposon jumping in and out of the pigment synthesis gene. And you can see that the Y kernel, the gene is inactive. So during kernel development, the transposon could jump out, so the gene becomes active again and start to make the pigment in the sector of the kernel surface. And you can also notice that this is a very active process, right? The transposon seems to be jumping a lot in all the kernels. This is another example that most likely is caused by transposon insertion, and that subject the gene to the DNA methylation control, and so that a sector of the surface, the PO cell, has more active gene transcription, while the others remain like the normal apple. There's another famous example is the agouti mice. So this strain of mice is due to a T transposon insertion into the promoter of the agouti gene, so that the gene now is regulated by DNA methylation of the transposable element. So what happened is these are genetically identical two mice. What the difference is, during pregnancy, during their mother's pregnancy, the diet rich in folic acid, then you tend to have a more normal DNA methylation, and that produces a normal looking more brown color regular mice. But if your diet is deficient in folate, and the gene methylation is not complete, so you get a more atopic expression of the agouti gene, and that disposes the mice to more obesity and other chronic disease. And there's another example for the nutrition regulated epigenetic is the queen bees versus the worker bees. And the main difference is because the queen bee were fed with royal jelly, so that also has a big effect on the methylation of the genome during their development. So this is a new variety of petunia that was recently, in the last few years, introduced into the market. And people speculate, so the underlying mechanism hasn't been fully discovered, but people speculate this is most likely due to a charcoal synthase gene cluster, and there is a transposon jump into one of the copies, and when the environmental condition, particularly when the flowers are developing, you need to have a colder temperature at night, and that triggers the expression of part of the gene that causes a double-stranded RNA and the RNAi pathway, and that shut down the gene in this beautiful sparkling patterns. Another good example is during the World War II, the Dutch hunger, because of a German blockade of the Netherlands city. A lot of children, pregnant women at that time, because they suffered to famine, so their children developed very under the malnutrition condition, and that affected a lot of imprinted gene methylation status. And what's more striking is now these kids are at their 60, 70 years old. They have their own second generation, and that disease is transmitted to their F2 population. So that's the area of imprinting regulation is very active right now. So my lab studied gene imprinting in plant. So imprinting is another strange example of epigenetics. So normally in the regular diploid cell, both copy, especially in Arabidopsis, it's a self-in organism. So paternal maternal copy are identical. Yet there are a small group of genes, roughly about 1 or 2% of the gene. They are monoallelically expressed in the endosperm. So that's compared to all the other genes. They are either on and or off on both copies. But this group of genes, only one copy is expressed, the other is silent. So how does a cell know which copy to maintain active, which one to maintain silent? Is quite interesting. Turns out that this has to do with the imprint that get established right before the two genomes comes together. So our research focused on this particular area of imprint establishment. So why imprinting is important? So there are many examples in mammal and also in plant that reproductive balance, reproductive success require the balance between this paternally expressed versus maternally expressed gene. And if you tip the balance, then the fetus isn't developed normally and prone to a lot of developmental disease and also throughout their adulthood. We study this process in plants. So you probably remember double fertilization is a unique process only occur in flowering plant. So during the fertilization, the pollen gland bring in a sperm cell, two sperm cells, and one of them fused to the egg cell, and that develop into the embryo. The other one fused to the central cell, and that develop into the endosperm. So central cell is special because it's a fusion of two polar nuclei right before fertilization. So the central cell is diploid, and after fertilization it's triploid. And endosperm mainly account for most of the human and animal food in the whole world. So in plant, gene imprinting only occur in the endosperm. There are a few examples of transient imprinting in the embryo, but it's not, it doesn't persist. So it can erase later on during embryo development. But in endosperm, the epigenetic asymmetry persists throughout the seed development. So, but the imprint is actually established right before the genome comes together. So gene imprinting is essential in plant. This is the first imprinted gene discovered in our plant, in Arabidopsis, and it was named by DEMETER. Sorry, it was named Medea because Medea killed her own child. So in this gene mutation, if you have a female mutant allele that's transmitted through the female gamete, then this seed will die. So in the heterozygote plant, you get a 50% aborted seed and 50% seeds are normal. And this mutant allele has no effect if it's coming from the pollen. And part of the reason is because the gene is required in the female gametophyte. And also it's because the male allele is not expressed during endosperm development. So if we look at, so this is a maternally expressed gene. So if we look at the expression pattern, it's turned on right before fertilization occurs, so in the central cell. And once it's turned on, the expression persists during endosperm development. But in order for this to be turned on, it needs the DEMETER gene. But if you look at the DEMETER expression, it's only on in the central cell, it's not on in the endosperm. So DEMETER is like the switch that we talked about earlier. Its main function is in the central cell, switch on the expression of the Medea gene, and once it's on, it continues to be on. But if you don't switch on, then it's not expressed after fertilization. So DEMETER regulate this Medea gene expression by epigenetic means. Just like the Medea gene, DEMETER is essential because it regulates the Medea. Medea is essential. So in the DEMETER mutant, you get a 50% aborted seed. And mainly the reason is because you're not turning on the polycomb gene Medea. Okay, DEMETER. So after many years of research, we finally established that DEMETER is actually a demethylase gene. It has a glycosylase domain, and you probably remember the function of glycosylase gene is to remove the mismatched base after the cell division or during when the DNA get damaged. And in this case, DEMETER recognizes a methylated cytosine and it chop off the base, and then there are other subsequent gene from the base excision repair pathway comes in to fill in the gap with unmethylated cytosine. So that's the mechanism of active demethylation in plant. That's a little bit different from in animal system. So many years ago, we established based on both in vivo and in vitro evidence, we're pretty confident that what happened in the central cell is DEMETER comes in, it gets turned on, and remove the methylation on the mediate gene and turn on the mediate. And this process doesn't occur on the sperm side. So when the two genomes comes together, the maternal allele remain hypomethylated compared to the paternal genome, and it's expressed, and the paternal genome is not expressed. So that seems to be the paradigm of plant gene imprinting, particularly for those maternally expressed genes. So and at that time there was a handful of example imprinted gene that seems to be regulated this way. So we know that DEMETER is a master regulator of gene imprinting, but we don't know how many more genes it regulates. So to answer that question, and partly because the genomic tool, the high throughput sequencing became available at that time. So we saw that in order to understand what DEMETER does in this cell, we cannot easily isolate this cell. This cell is embedded in a lot of other maternal tissue that's within the female carpel, one of the ovule, and that's also embedded in the unopened flower buds. So it's even until now, it's difficult to purify pure enough central cell population for more detailed genomic work or biochemical work. So we use endosperm and the embryo kind of as a proxy, because the demethylation only occurs in central cell that become the endosperm, but it doesn't occur in the egg cell, so that becomes the embryo. So we can compare embryo and endosperm, try to figure out where the demethylation occurs. And we can also look at maternal allele, paternal allele within the endosperm. Use that to try to delineate where the target sites are. So this was done by a bunch of high throughput study, genomic study, and we came to the more clear picture. DEMETER actually demethylate two cells. They are called the gamete companion cell, the central cell and the vegetative cell. So central cell is on the female side, vegetative cell is on the male side. So this is a mature pollen grain. You have a vegetative nucleus, and the two sperm cells are embedded within that vegetative cell. And this pollen will germinate and form the pollen tube. So part of the function of a vegetative cell is to facilitate the pollen tube grow and to help with delivering the two sperm cells to the female gametes. So from the genomic work, we know that DEMETER mainly demethylates short transportable elements that flank the coding sequence, and because of their demethylation that affect the transcription of this gene nearby. It also demethylates a lot of intergenic and heterochromatin sequences for reasons that we don't quite understand yet, but there are some hypotheses. So on the female side, it established gene imprinting in the endosperm, and on the male side, and also on the female side, most likely the hypothesis is it demethylate the transposon and activate the transcription of the transposable element for the purpose of producing the small RNA, and then small RNA get transported to the egg cell. So I can summarize that in the next slide. So what we learned so far at that time was DEMETER's function isn't really for the regulating gene imprinting, but for a bigger picture is try to maintain the transgenerational epigenetic inheritance. So this is just an example on the male side. DEMETER remove the methylation, turn on the transcription of the transposon, produce small RNA. This small RNA get shuffled to the sperm cell, and for the purpose of maintaining the methylation of the sperm. So if we have a mutant allele in pollen, and we can actually see that if this demethylation doesn't occur, the sperm methylation actually decreases. So the function of demethylation here is to reinforce the methylation in the sperm cell. Why would you want to do that? If you turn on the transposon, we know that transposon will start to jump and activate, and then that kind of compromise your epigenome integrity, right? So it's actually harmful for the cell. That's why the cell evolved all the methylation pathway, try to silence the transposable element. But because the vegetative nucleus, and also on the female side, the central cell nucleus, the genome is not transmitted to the next generation. So that's part of the theory that this genome sacrifices itself in order to reveal where the transposons are that need to be reinforced for the next generation. And what happened to imprinting? So we know that transposon come and goes, so it jumps, and then by chance it jumps to near the promoter of whatever the given genes, and then it got caught in the male, especially in the central cell, by this global demethylation process. And because of that, and it gets turned on the female side. So gene imprinting, you can think of it as kind of a byproduct of this global demethylation event, or maybe it's just got co-opted. The mechanism just got co-opted by the endosperm in order to use it for the gene imprinting control. Okay, so we know based on the genomic work, we know where DEMETER demethylate. And we look at the genome browser by comparing embryo, mutant endosperm, and wild-type endosperm, then we can pretty much see where it acts. And turns out that, just like I said earlier, it demethylates a discrete genomic loci. So it doesn't touch the nearby methylation, DNA methylation. So there is some sort of a specificity, a targeting mechanism that is directing where it needs to go. So we are very interested in trying to figure out what that is. So that's, I think this is probably the very first experiment our lab did when I joined the NC State. So that's been a while. So we can talk to Changqing and Yu Hong will tell you a lot about the struggle that we went through trying to figure out exactly what it means. Okay, so DEMETER is a glycosylase protein, but it's only a small glycosylase domain embedded in a very long polypeptide. So that's about 2,000 amino acids long. And there are several conserved regions and then variable regions. So in early days, people started to do deletion and to look for enzymatic activity. If you delete into the A region or into the V region, you lose enzyme activity in vitro. So we know that this part is critical for the enzyme. So we always call it a catalytic core. So in our picture, in our mind, this is just an enzyme. So whatever is taking the DEMETER to where it needs to go must be conducted by the N-terminal region. So we saw that well, what if we chop off the N-terminal region and see if that gene can complement? So there's a basic region that used to, they've been shown to encode the nuclear localization signal. If we chop it all out, then this C-terminal region doesn't work. We add in a nuclear localization signal and controlled by a DMETER promoter, and we put it into the mutant plant. So in the homozygote mutant, 99.9% of the seed die. We put in a copy, then it recovers to 50%. We put in, when the transgene is homozygous, then it fully restored to the normal seed. So C-terminal is sufficient. That was a big surprise to many of us. And we look at a few candidate gene, target gene, it restored the expression to close to wild type level. So we then look at, okay, so there's another function that's DMETER is absolutely required for the female, on the female ovule. If you have a mutant, then this seed will abort. Now, if your female is wild type, and on the male side it's heterozygote, even though the DMETER has no function here, in the endosperm, the paternal allele has no function, it affects the pollen transmission. So you're supposed to get a 1-to-1 ratio if it transmit randomly from the pollen. You should get 1-to-1, but instead you get a almost 4-to-1 ratio of wild type versus heterozygote mutant. So we asked whether our C-terminal construct will also complement this defect. So now, if you have a homozygote pollen and then one copy of the transgene, and now we found that instead of 50-50 of all the mutant got transmitted equally, the pollen that has a transgene, it actually gets transmitted more effectively. So we can conclude that the C-terminal transgene complements the female side and also the male side defect. Okay, so now next thing we want to know, what else does it do in the central cell? So again, we take the advantage of the endosperm as a proxy. So we can actually compare mutant endosperm that in theory the target demethylation doesn't occur in mutant. And then we can look at the rescue endosperm, what happened to the methylation status. So this gene is important for gene imprinting. So we look at the known target genes and where the demethylation occur. And we know that from earlier study, we know where they are. And sure enough, the C-terminal also demethylate, but just not as much as the wild type. So that's, in a way, that's expected, right? You only use a half of the protein, but the fact that you know where to go is still pretty amazing. So we suggest that the C-terminal alone is sufficient to whatever the in vivo function that the DMETER does. Okay, if we look at the methylation profile in each target loci, so we can compare the mutant that should have a high methylation and the wild type, and then we look at the differences. And if the difference match the threshold that we define, then we call it the target gene. We know that the DMETER goes there to remove methylation. So from comparing the mutant and the wild type, we know where the target genes are. And we do the same thing comparing mutant and the mutant that has been complemented by the C-terminal, and we know where they are. So we try to compare these two groups of the loci to figure out if there are any difference. Well, turns out they partially overlap. Okay. But they target about the same number of the loci, so roughly about 10,000 target sites that we can identify. But that's it. That's not very telling, right? You're not using the same protein, so of course they behave differently. So that took us a very long time to go from here to the next step. So we want to see why these target sites are missed by the C-terminal. Are they really missed, or what happened there? So when we deal with this large data set, genomic data set, we use several different analysis, and each one tells us slightly different things. We kind of have to paste the information we got together using a multiple analysis. So one way to look at to compare the two genomes is we use a lot of a kernel density plot of the methylation differences between two genomes. So if there's no difference in these two genomes, you subtract the methylation level, and that gives you a very nice sharp peak close to zero. That tells you there's no difference. But if the peak gets shifted to either side and the shoulder becomes broader, then you know there's something going on. Especially if the major peak shifted one way, then that signals a very global methylation differences between these two genomes. So we use this method to come back and look at this region that got missed by the C-terminal. Okay, and we ask, is it really missed, or does the C-terminal demethylate this region at all? So these are the endogenous DMETER target sites. So the methylation difference is pretty big, and that's reflected with a dotted black line here. Now, if these sites are not demethylated at all by the C-terminal, then the difference between mutant and this red bar should be zero, but it's not. It's shifted to the positive side, so that means it is demethylated. They are demethylated, but just not to the same degree of wild type. So that falls below the criteria that we would call it a target site. Okay, but still, that's also not enough. What does it mean? Why it doesn't demethylate well enough? So this is one advantage of working with this model plant is there are a lot of resources available. So this has been looked at. Arabidopsis whole genome has been categorized into 9 different chromatin states based on all the genomic features, including the various kind of histone modification, gene density, CG content, gene and TE density, etc. So state 1 and 7 are euchromatic sequence. They are more open, more accessible. Heterochromatin are state 8 and 9. Okay, so now we look at this region number 3 that got missed by the C-terminal enzyme. You can see that it is very enriched for the heterochromatin state number 8 and number 9. The C-terminal also has its atopic site, and the region 1 is very enriched for the euchromatic features. So it seems that now if we compare to all the target by the endogenous DMETER versus all the target by the C-terminal, the endogenous target has a lot of heterochromatin region, and this got heavily reduced but enriched for the euchromatic site. Okay, so another way to look at the target site in endogenous target, we group the target site based on their length and the overall the base pair it covers. So DEMETER is roughly about close to 4 million base pairs regions, and then a lot of the target sites are longer than 1 kb, especially 1 kb and 1.5 kb. If you look at the characteristic of these longer target sites, they are almost exclusively heterochromatin regions. Now, the same analysis shows us the C-terminal doesn't have those long targets. So it's telling us that these are long heterochromatin target sites are either missed or not demethylated very well. So we look at these 250 longer target. Actually, most of them are also targeted by the C-terminal, but their average length is just very small. Okay, so this is, you can see that in the genome browser. These are the wild type target sites. They are longer, but they are also targeted by the C-terminal. It's just very short. Why is it very short? So the DNA is wrapped around the nucleosome, and between the nucleosome is bound by H1. And so these are the more open chromatin region, but in the heterochromatin region, they are further compacted into a more condensed structure with the help of the H1 linker histone. So it's known for a long time that H1 and this nucleosome are barrier for transcription factor or DNA modifying enzyme. In the case of DNA methylation, methyltransferase, you need another chromatin remodeling complex to temporarily disrupt this compacted nucleosome structure in order for the enzyme to get access to. We think that this might be what happened to the C-terminal enzyme. So turns out that this is a complex called facilitated chromatin transcription transaction complex. The FAC complex is required for DME demethylation. That was shown a while ago by a genetic study. So if you look at this wild type with the target gene, it's all expressed in the central cell and after fertilization in the endosperm. But if you have a mutation in one of these components of the complex, then it doesn't turn on the target gene. So, and then later on, about a year ago, large-scale genomic study showed that about half of the DEMETER target site actually require this FAC complex, and they are all in the heterochromatin region. So what does it do? The FAC complex, in this case of transcription, the transcription machinery cannot get through the nucleosome template, so it needs a FAC to temporarily remove the nucleosome, and then so that the transcription will go through, and then it reassemble the nucleosome back after the transcription. Okay, so we looked at what happened to the. So these are the endogenous DEMETER target, and part of it, about half of them need a FAC complex, the other half doesn't. So we group them into a dependent and independent loci. So you can look at the kernel density plot. This direction means more demethylation by the enzyme. In a wild type, both groups are demethylated to the same degree, dependent and independent, in a wild type endosperm. In the C-terminal endosperm, independent loci is slightly less, but the FAC-dependent loci is much more. severe than the independent. So based on this, we think that the C-terminal enzyme behavior could be due to lack of FAC activity. So the C-terminal region is missing the whole, almost half of the protein in the N-terminal. So we suspect that the conserved region at the N-terminal is most likely mediate the recruitment of the FAC complexes for this heterochromatin target site. The C-terminal alone is functional, but why do you need all this? And perhaps this represents a prototype of the DNA demethylates. So we did an evolutionary study, and we looked at all the land plant lineage. We found that this N-terminal conserved region only present in the angiosperm. It's not present in the non-vasculature. So that showed us, that tells us that yes, indeed, the C-terminal version of the domain architecture most likely represent a prototype of the demethylates. And the N-terminal region is acquired during angiosperm evolution, and which incidentally, angiosperm is where the gene imprinting occurs. So acquiring the N-terminal region may directly or indirectly involve in the evolution of gene imprinting regulation. Okay, so based on our result, this is our working model. We think that the DEMETER has two separate units. It's a bipartite organization, and the C-terminal direct where it needs to go. And when it encounters the nucleosome, and then the N-terminal recruit the FAC complex to remove the nucleosome so that it can process the longer target demethylation. In the C-terminal version of the enzyme, it also knows where to go, but it gets stuck. So when it encounters the nucleosome, then it doesn't move forward. So we think that's, at least that's the data is telling us. What about the ectopic side that I haven't talked about? Okay, remember that the two target circle doesn't overlap. So what happened here? So here we know that they are enriched for the euchromatic region. Again, now using the kernel density plot, we were asking, so these are the target site for the C-terminal. Does it get demethylated at all by the endogenous protein? So the peak is close to 0, but it has a broad shoulder. So some site within here are also demethylated by the endogenous DEMETER. So we further separate these loci based on their genome feature. So the green, so the genic, the longer and the shorter TE, they all get demethylated a little bit by the endogenous protein, but the coding gene is not. So that tells us that the C-terminal DEMETER is atopically demethylated these coding genes. So if we look at all the coding genes, some are demethylated by endogenous protein, and then a lot of them are atopically demethylated by C-terminal region. So the coding gene methylation tend to be the genes that are under active transcription. Indeed, we see, yes, all these new atopic demethylated genes tend to have a higher expression in the endosperm. So we think one function of the N-terminal region is to restrict the DEMETER from targeting these coding sequences, but we don't know what kind of mechanism yet. Okay, so this kind of a summary slide. We think the data is telling us that C-terminal is directing localization of the DEMETER. And if we look at the conserved region within the C-terminal, it's mainly the glycosylase domain. And then there are two interesting domains, CXXC, and then the RRM, the RNA binding motif. Both of them have been implicated in epigenetic regulation. The mammalian CXXC it's a thin finger protein that can distinguish methylated versus unmethylated cytosine. So we think that this most likely is at least one part of the major mechanism of directing the protein. The RRM is known to bind a non-coding RNA. So non-coding RNA could also be a targeting mechanism that bring the epigenetic complex to whatever the target site by matching the sequence. But I'm just a little skeptical because if you look at the RNA motif among different genes, the meter-like gene, they are just too conserved. So we don't know, but at least we know that this motif and this domain could likely be the key function in directing the meter regulation. So with that, I want to acknowledge the people in my lab, Changqin and Yuhong, are mainly responsible for this work that I described. And with the help of our collaborator in NCBI, St. Louis University, that helped us with evolutionary study. UC Berkeley team recently published the FACT genomic work that give us a great foundation to figure out what our data means. Jin in South Korea, his lab was actually the first one to find C-terminal region might be sufficient for in vivo function. So the funding is from the Hatch project and also from NSF grant. Okay, thank you for your attention.